Friends, welcome to Biology Made Easy. Today in this video, we are going to discuss the supplementary genes. Supplementary genes, and it gives a ratio of 9:3:4. So this ratio always you have to remember because that is very important for examination point of view. Now what is a supplementary gene interaction? Like a complementary gene interaction, this reaction is also non-allelic. That means there are two independent pairs of genes. They are non-allelic. That means they may be present in different loci of the same chromosome or may be present in different chromosomes. But that means they are always non-allelic. That means two pairs of genes which are completely non-allelic, out of which one dominant gene expresses itself and produces some character. But the second one, when present alone, does not produce any character. But when it is present along with the first one, it enhances the character of the first one, producing a new trait. So, if we'll write it in the form of definition, there are two independent pairs of genes, in bracket I am writing non-allelic, of which one dominant gene expresses its character, but the second one does not express when present alone, but enhances the effect of first gene to produce a new trait. So this is definition. Now let us take some examples. So if we take the example number 1. So the example number 1 is, we can take that of the coat color of mice. So in mice there are different colors, like they may be black, they may be white, they may be agouti. But how these colors are produced? Let us see. Suppose the gene B is present in dominant form, then it produces black color. If it is present alone, then there is the dominant gene A. This no character. That means when it is present alone, it doesn't produce any character, but enhances the effect of gene A when present together. Now, when both A and B are present together, it produces a new character, that is agouti. And when both the dominants are absent, that means they are present in the lower case, then it gives rise to a color, that is albino. Albino means a pure white. So in case of a coat color of mice, these are the different genes and their expression. Now, if a cross is made between the black and white, a cross is made between a black mouse with a white mouse having the genotypes. In black case, it is capital B, small a, and in case of white, it is small b, and capital A. These are the parents. Now, what will be the hybrid? The hybrid will be capital B, capital A, small a. This is the F1 hybrid, and the F1 hybrid, it becomes Agouti. Because both the dominant genes A and B are present. In the F2 generation, what is happening, you see. When this plant will be selfed, that means we'll make a cross between capital B, capital A, small a, with another one, capital B, capital A, small b. When selfing will be done. So what will be said? The genes which will be produced, if we will draw the Punnett square, so in the Punnett square, you see, this will be capital B, capital A, this will be capital B, small a, this will be small b, capital A, and this will be small b, small a. Similarly, this will be capital B, capital A, then capital B, small a, then small b, capital A, and small b, small a. So, in this case, what is happening? Capital B, capital A, capital A. Here capital B, capital A, small a. Here capital B, small b, capital A. And here capital B, small b, capital A, small a. In this case, capital B, capital A, small a. Here capital B, small a. Here capital B, small b, capital A, small a. And in this case, capital B, small b, small a, small a. Now here it will be capital B, capital A, small a. And in this case, capital B, capital A, small a. And here small b, and capital A. Here small b, capital A, small a. And in this case, it is capital B, capital A, small a. And it will be capital B, and small a, and this one will be small b, capital A, small a, and this will be all four in the lower case. So in this case, what is happening, you see. Now in this case, both the dominant genes are present in how many cases, see. Now here capital B and capital A are present, that means all these, in all these four, and here capital B, capital is there, that means in this row also all. Similarly, here capital B is there, here capital A is there, this one, and here capital A is there, here capital B is there, this one. So now you count through how many tick marks are there. 1, 2, 3, 4, 5, 6, 7, 8, 9. That means in these 9 cases, in the 9 by 16 cases, the color will be Agouti, because both the dominant genes are present. That is Agouti. Now you see, in this case, what is happening? Here it is only dominant gene B is there, A is not there. That means this will be black in color. Here only dominant gene B is there, this will be black in color. And here also only dominant gene B is there, so this is black in color. So how many black are there? Now you see 3 out of 16 are black. Then you see these cases. Here you see the B is present in a recessive form. Here also it is present in recessive form. Here also recessive form, and here also it is present in a recessive form. That means here B is present in recessive form, which is responsible to produce the black color if it is present in a dominant gene. And A is there. A is unable to express when present alone. So here also capital A is there. It cannot produce any character. And here also it cannot produce any character because B is present in recessive form. And here there is no dominant gene. All are in the lowercase. So in these four cases, where only B is present in recessive form, they are albino. So these are all albino. That means the 4 by 16, they will be albino. That means the 9:3:4, this is the modified ratio of 9:3:3:1. So in this case, these 3 and 4, they are combining to give rise to this 4. Because here you see the gene, when it is the gene B, when present in dominant form, it is producing black color. But when it is present in recessive form, it is unable to produce any color. That is why in all these 4 cases, the color is albino. So here what is happening? Now the B gene, when present in dominant form, it is expressing a character, and gene A, when present in dominant form, is unable to express any character. But when both are present together, the gene A, which is unable to express itself when present alone, it is enhancing the effect of a B so that a new trait is produced. That means here the action of A is supplementary to the action of B. That means here we can tell that both the dominant genes B and A are supplementary, giving a ratio of 9:3:4. So this is one of the examples. We can also give a second example. The second example is development of grain color in maize, which is regulated by 2 genes. The development of grain color in maize. So the example number 2, development of grain color in maize. So here the development of grain color in maize, it is regulated usually by 2 genes, 2 dominant genes, R and P. Now when R is present alone, it produces red color. When P is present alone, it has no expression. But when it is present along with red color, it enhances the color into purple. And when both are present together in the dominant form, it is giving a color purple. And when both all are present in a recessive form, then the color is usually white. So when a cross is made between purple and white, so the purple is capital R, capital P, and the white is small r, small p, and the F1 hybrid will be capital R, capital P, small p. And here both are present in dominant form, so naturally this will also be purple, like this one. But what is happening in the F2 generation? Now you see the F2 generation, this will be capital R, capital P, this will be capital R, small p, then small r, capital P, small r, small p. So this side also capital R, capital P, capital R, small p, small r, capital P, small r, small p. So if you draw like that, we'll find, I am not drawing, you can draw the whole thing. Here capital R, capital P there. That means in all these four cases, there will be capital R and capital P. Here capital R, capital P is there. That means in this column also, all will be capital R and capital P. Then here capital R is there, here capital P is there. So here both the dominant genes are there. Then here capital P is there, here capital R is there. This. Now you see the tick mark is at 9 places. So at these 9 places, both the dominant genes R and P are present. So when both are present, it will give a ratio of 9 by 16, and this 9 by 16, they will be purple because both the genes are present. Then you see in how many cases only this capital R is there. So only capital R is present here. In this case, you see it is capital R and small p, small p. And when only capital R is there, it is red in color. That means this will be red in color. Again you see, here also it will be capital R, small r, and small p, small p. So here capital R is there, and this will also be red in color. This is red in color. And here you see, this will be capital R, small r, and small p, small p. And here capital R is there, so this will also be red in color. That means this 3 by 16, they will be red in color. Now see in these 4 cases. So in these 4 cases, so what is happening, you see? So from this side, small r is coming, but the P is present in dominant form. And here the R is in lowercase, and one P is dominant, the other is recessive. And here the R, both they are in lowercase, and one dominant gene P is there, the other is in lowercase. And here all the 4 are in lowercase. And again you see the dominant gene R, which is responsible for production of the purple color, that is present in lowercase. So here you see smaller, here smaller, here smaller, here smaller. That means in these 4 cases, the gene R is present in the lowercase. It is present in the recessive form, and it cannot express its character. That is why at these 4 places, the color will be white. That means 4 by 16 will be white. That means the F2 generation ratio, is 9:3:4. The 9 are purple, and these 3 they are red, and these 4 white. As in the that case, as in case of that lateral, here also the same thing. So in case of supplementary gene interaction, one gene expresses its character, but the other gene, it doesn't produce its character. But when it is present along with first, it enhances the effect of the first one. So this the interaction between them is usually supplementary. That means here the gene P, when present alone, is unable to express, but when present along with the R, it is enhancing its effect. So this is about the supplementary gene interaction. I hope you have clearly understood the thing. Thank you very much. God bless you. Have a good day.